Let us discuss about Hershey and Chage experiment. 1952, Alfred Hershey and Martha Chase provided convincing evidence that DNA is genetic material. Wiring blender experiment using T20 bacteriophage and bacteria. Radioactive levels P32 for DNA, S35 for protein. These are the pics about Hershey and Chase experiment. See, it was labeled as S35. Now T2 bacteriophage is attacking on Escherichia coli. And finally, most of radioactivity recovered in phage ghost. See, this is the DNA inside the cap of T2 bacteriophage P32. Now attacking the bacterium, and finally most of the radioactivity recovered in bacteria. Finally, Hershey and Chase experiment. It was performed in 1952 using bacteriophage, a type of virus that have a very simple structure, an outer core and an inner component. The phages are made up of equal parts of protein and DNA. It was known that the phage infect the anchoring the outer cell to the cell surface and then deposit the inner component to the cell, infecting it. Scientists were interested in finding out whether it was the protein or component or DNA component that got deposited inside the infected cell. By incorporating radio level either in protein or the DNA of the infecting phase, they determined that the DNA was indeed introduced into the infected bacteria, causing proliferation of new phages.